All right. Hello, everyone. Thanks for joining us today for this Taudia webinar today. My name is Chris Ng, Director of Operations at Taudia, and I will be your moderator today. In this webinar, Dr. Jennifer Yokoyama and Taylor Johnson from University of California, San Francisco will be discussing dried plasma spot biomarker test for Alzheimer's disease population screening using qPCR systems. During the presentation, if you have any questions, you can submit them using the Q&A tab at the bottom of your screen. Please put your questions here rather than in the chat window so we can record your questions to answer during the Q&A session following the main presentation. As a reminder, we are recording this webinar, so if you miss any part of it or would like to share it with your fellow colleagues, you will be notified in a few days via email that the video is ready. Without further ado, I will hand it over to Dr. Yokoyama.
Morning, everyone, and thanks for coming. I'm going to just take over Taylor's slides momentarily. We're going to do a little tag team here. I will lay the foundation a bit for what we're going to be talking about today, and then Taylor will be the meat of our sandwich and tell you about our experiences leveraging dried plasma spots for doing some biomarker measurements. But first, just to lay the foundation. So these are mostly funding sources, not relevant to the talk today, although I will note that I am on the advisory board for Taudia, and this mainly reflects my belief in their technology. So my name's Jennifer Yokoyama. I am a neurogeneticist, and I run a research lab at UCSF in the Fine Memory and Aging Center. And our lab is really focused on trying to utilize genetic variation to understand how there are differences across populations for both risk and resilience to diseases like Alzheimer's disease and frontotemporal dementia. So these are neurodegenerative diseases that can result in dementia syndromes. And our lab really approaches these diseases from a clinical research perspective, and we do leverage genomics, transcriptomics, and neuroimaging methodologies, mainly both to gain an understanding of the biology of disease, as well as to try to identify new biomarkers and therapeutic targets that we might be able to utilize to reduce the burden of neurodegeneration. And so just for those who are less familiar with Alzheimer's disease, this is a neurodegenerative disease that results in a dementia clinical syndrome. And I'm going to refer to this class of disorders or clinical syndromes as Alzheimer's disease and related dementias. So many of you are probably familiar with AD or Alzheimer's disease in the context of older adults who have memory changes, can get confused easily. And we know that the burden of Alzheimer's disease is increasing as we are basically extending our lifespan. So while overall this is not a terrible problem to have that we're living longer, the reality is that while in some regions of the world the burden of Alzheimer's disease is going down, on average across the globe, this number of individuals affected is continuing to rise. And in fact, the cost of dementia care, this is an estimate in the United States, but when you extrapolate this across the world and thinking about the potential burden over time, this is quite tremendous. And this is not just the cost of clinical care, but it is also the loss of employment dollars in the context of the individuals who are care partners of those living with dementia, as well as the ecosystem or infrastructure of the health systems that are basically trying to cope with this change in disease. And so, obviously there is strong motivation for us as a field to try to reduce this burden of Alzheimer's disease. And the reality is, as I mentioned, that for some populations or subgroups, there is a reduction of Alzheimer's disease risk occurring. However, on average, the burden for ADRD is by far the largest in women and in people of color and those with lower incomes, as well as those in lower or middle income countries. And so, we know that not just because women on average or females tend to live longer, but for other biological reasons as well that we're still trying to understand, that the burden of risk is quite larger in women who are also on average the typical care partners for those living with dementia. And of course, as I mentioned, that there's this increased burden of dementia in individuals of color. And this is something that we see in particular in the United States because of differences in access to care, as well as where people are living in the country, and other reasons. But regardless, this means that there is a increase in burden in these participants and individuals who tend to be those who are less well characterized in research. So quick primer on the Alzheimer's disease biology. Don't worry, there will not be a quiz after this webinar, but just as a reminder. So the way that we characterize our neurodegenerative diseases is through the proteins that aggregate and cause damage to the neurons or the cells of the brain in the context of disease. And so for Alzheimer's disease, the two main proteins that we're interested in are amyloid, which causes these abnormal plaques around the cells of the brain. And my slides keep moving, and I'm not sure why. And also tau, which on average, when it's healthy, helps stabilize the long parts of neurons by stabilizing microtubules, but in the context of abnormal pathology, can fall off those microtubules and cause tangles in the neurons themselves. And this tau tangle or neurofibrillary tangle results in the death of the neurons, which ultimately results in neurodegeneration. So these are then basically the cells that are responsible for signaling in the brain breaking down, and that, as a consequence, results in the cognitive decline that we see associated with the dementia clinical syndrome. So here's a loss of brain matter or atrophy of the brain and a consequential cognitive decline, often manifesting as changes in memory, but can sometimes also manifest as changes in language, as well as executive function or the ability to basically do multitasking. And so as a consequence, there's a lot of confusion, a lot of forgetfulness, ultimately an inability to complete daily tasks, and occasionally, or actually relatively often, also mood swings. And so you can imagine if this type of clinical manifestation is occurring and building up over time, this can increasingly cause challenges for the family members and friends and care partners of those living with dementia. And so there's a move in the field to really leverage the fact that we understand the trajectory of the biology of disease. And our lab, like many others, are anchored in this ultimate goal of trying to reduce the burden of disease. This is our value framework that we use in the Yokoyama lab. Different labs have different strategies. Some are really looking for cures or preventions for Alzheimer's disease. For us, we're thinking both of ways that we can understand the disease biology, but also to reduce the burden of disease by identifying those at risk earlier in the disease process. And so that really relies leveraging our understanding of that disease biology that's occurring even before all of those clinical symptoms are manifesting. And one of the ways that we can do that is by leveraging diverse populations. And we know as geneticists that there are many strengths for studying diverse populations. From a genomic perspective, we can leverage the fact that there are differences across the genomic ancestral background of individuals from admixed or diverse populations. And because of the migration patterns of human populations over time, this allows us basically to better leverage differences across people's genomes to identify new genes that are underlying or associated with disease risk. They can improve our ability to reduce the amount of genomic space that might be contributing to a specific disease. And importantly, it allows us to better estimate the effect of any genetic risk for disease that is very specific for a given population based on their background ancestry. So, for example, for me as an East Asian, I'm Chinese and Japanese American. And so for me, my risk for a given genetic variation in an East Asian genome might be slightly different than, for example, Taylor's, who will be talking next and is coming from a non-Hispanic, white, or European background genetically. And so because of this power of population genetics, we are part of multiple consortiums, including this Asian cohort for Alzheimer's disease led by Lee-San Wang at the University of Pennsylvania, as well as many colleagues around North America to study what is actually the first large cohort of Asian Americans and Canadian Asians for Alzheimer's disease. In addition, I am part of the team of individuals we've actually just received a renewal for the Red Lat project. This is a US Latin American project led by Doctors Agustina Banez and Bruce Miller to study genetic neuroimaging, cognitive behavioral correlates of Alzheimer's disease and frontotemporal dementia, and then try to understand how the social determinants of health impact all of these clinical measures of dementia in more diverse populations, largely from South America. And so again, we are really trying to leverage these differences, not just in genomic population diversity, but also in social and lived experiences to better understand how these diseases manifest in the brain in different life situations. And the reality is, a lot of this work is pioneered by members of our Global Brain Health Institute who participated as Atlantic Fellows for Equity in Brain Health. And I'm a faculty member of GBHI. And this is just a map that's actually now outdated, showing you some of the breadth of the regions where we now have fellows who are interested in leading efforts to improve brain health and reduce the scale of dementia impact around the world. And so many of our fellows are researchers who are clinicians who are trying to advance their research and work in the field or locally in their clinics with their patients. And the challenge remains that for Alzheimer's disease, we are now in a space where we have so much, leveraged this biological understanding of disease that it's now becoming almost a requirement to have biomarker evidence of Alzheimer's disease dementia and Alzheimer's disease biology for publications. And so this here, for those in the Alzheimer's disease field, is very familiar. For those who are not, this is a curve set by Cliff Jack and colleagues, which basically shows how the different biomarkers for Alzheimer's disease change over time. And so just quickly, we see basically that changes in amyloid actually show up really early, up to 20 years or more before any clinical symptoms of Alzheimer's disease, the dementia syndrome, show up. Next, we have changes in tau neuropathology. And then finally, there are changes in the neuronal density or neurodegeneration. And then because of that neurodegeneration, there are clinical changes. And so while the majority of observational research has really leveraged the fact that we can measure clinical symptoms in patients, the reality is the rest of the ADRD field is really now moving into this pre-symptomatic phase where we are defining having Alzheimer's disease as being positive for the protein biomarkers underlying disease. So this means that those who do not have access readily to these types of biomarker measures, which traditionally had been through expensive PET imaging and now more recently through biomarker measurements in cerebrospinal fluid or now plasma, are going to be at a disadvantage. So if we, for example, can't afford or have the infrastructure to measure biomarker levels of amyloid and tau in our neurodegenerative disease patients in Latin America, then we are going to be at a disadvantage trying to publish our papers on these patients who probably show the same clinical manifestation of disease in terms of the clinical syndrome, but we just don't have the biological tools yet to demonstrate that the disease biology underlying that clinical syndrome is actually Alzheimer's disease. And so this is why I was super excited to learn about Taudia's splash technology, because to me, this represents an excellent bridge into research opportunities for under-resourced populations. And so I'm not going to go through the chemistry in detail, but you're welcome to ask questions during the Q&A, and our experts at Taudia are happy to answer those. But suffice it to say, um, as a person who is used to studying molecules of DNA and RNA, protein biomarker work has sometimes been daunting because it's typically relied on expensive technologies or mass spec or fancy assays with a lot of antibodies and things I understand, but like on a practical level are not feasible for our lab. And the beauty of the Splash platform is that it actually leverages the tools that we as molecular biologists, geneticists, are used to using, which is PCR. And so by leveraging the fact that we have antibodies that recognize these protein targets really well, the Splash technology basically uses this information to have a very simple oligo or DNA-based readout that we can then measure on a PCR machine. The other beauty of the assay is it's relatively straightforward, and so it can be done in a relatively brief period of time. And again, it leverages the qPCR machine that basically every lab around the world has at our clinic for measuring COVID levels, for example, or every molecular biologist may have in their lab for running PCR and doing basic molecular screens. So I think this, for us, was really a unique opportunity to think about how we could leverage this type of technology to begin to address this gap in opportunities for under-resourced groups. And so what I did, as all PIs do, is I was like, okay, Taylor, my trusty lab manager, can you help us figure out if this is going to actually be feasible to do in the field? And so I'm going to hand it over to Taylor now, who is going to tell you about our experience.
There we go. Hi, everyone. My name is Taylor Johnson. I'm a staff research associate working with Jennifer Yokoyama. So thanks for that foundation. So given these challenges, we set out to design a workflow that would basically work to mitigate each logistical hurdle in the way of obtaining reliable, sensitive, and accurate Alzheimer's disease biomarker levels from remote participants. So standard procedures for biomarker detection, as we said, limits accessibility. These main points are due to lack of high quality biospecimen, which is sensitive to post-sample time interval, temperature sensitivities, and also highly regulated shipping regulations. There's also issues with biospecimen accessibility, which requires a mildly invasive procedure and also requires a trained phlebotomist to be on site. And as Jen mentioned, there's expensive instrumentation involved, which often requires specialized training. And so what this leads us to is uneven global coverage of Alzheimer's disease detection and treatment, limits the understanding of disease biology across ethnic groups, and restricts access to care near to central processing facilities. And the sampling constraints hinder longitudinal collection, ultimately for prognostic development. And so breaking down our workflow into these three main components, we're able to kind of pick at each point to optimize and put it back together to make this thing work. So we start with collection, move into processing, and then into analysis. And this goes from whole blood. We will be sampling into Capitainer Sept10 for a dried plasma spot, which I'll talk about in just a moment. There will then be the dried plasma spot elution, which we will run on the Taudia splash assay, and then that will go through our analysis pipeline. And if you're interested, we do have basically this first part of our workflow published on protocols.io, and we are working on getting all other protocols uploaded so that we can contribute to reproducibility at the bench. And so the Capitainer Sept10 kit for a dried plasma spot is really the first step in optimizing this workflow to increase the accessibility of biospecimen for the biomarker detection. And basically what it is, it's a card with two microfluidic channels that lead to a fiberglass disc that reliably samples 10 microliters of dried plasma. And this specific 10 microliter sampling is key to then quantifying our biomarkers downstream, as we're able to then design a dilution factor to then get back to our original concentration. And so in our space, we leveraged existing ACD yellow top tubes that we were using for PBMC isolations. We pipetted in 75 microliters of whole blood into each channel. And the way these cards work is whether it's a capillary lancet, it's from ACD, whole blood, or EDTA per standard collections. You will fill to that red vertical line, and then place the card under observation. There should be a blue dye that manifests to confirm successful sampling. And then what we do is we just store it at room temperature. And this card can be shipped, like I said, at standard temperatures without regulations. It can be just sent straight through the mail. And so that really opens up the door just for obtaining the samples in the first place. To then elute, we take off this dried plasma spot and we will elute in 200 microliters of a sample buffer, which creates a 20x dilution. That is then incubated for 45 minutes at room temperature under agitation. And then we will throw into the Taudia assay. And this solution is effectively just the straight up plasma that you would otherwise get from a normal pipeline. And so we were able to collect over the course of 34 days, roughly 1 month. A lot of these participants were either the first time coming into our clinic or were not yet able to acquire the appropriate diagnosis. So most of these participants were unknown coming into our lab. We were able to collect through the UCSF Fine Memory and Aging Center Parent Program Grant, Brain Aging Network for Cognitive Health, and Alzheimer's Disease Research Center. And so looking just plainly at the cohort, we were able to obtain a pretty even split of male and female. And I'd like to highlight the sample stability status rows here, where we had a pretty good split of samples that were at room temperature for less than 14 days and samples that were sitting for greater than 14 days. And so I think we're effectively capturing a pretty good range here that we should otherwise be able to see if there's a decline in quality of our analyte through the Taudia assay. And so before we get into the real data, I wanted to look at some quality control metrics. And so what we see here is on the left, we have p-tau 217, and on the right, we have A-beta 42, which will be useful in taking our ratio for our clinically relevant biomarker. And what we did is we plated a 1x and a 4x dilution of each sample. And we ran the Taudia assay on that, and what we were hoping to see is a roughly two CT shift across the samples and targets, which we did in 1.5 to 1.7. What this indicates is probe specificity, and that we aren't seeing any biases or effects of our dried plasma spot collection downstream in the assay. And so this is good news for the data that's coming out that we'll talk about in just a minute. And so here we have on the x-axis our 16 samples participants, and on the y we have a log scale amyloid beta 42. And the first thing I'd like to highlight is that we got sample out of these, which was very exciting, given that these cards were from 1 to 34 days effectively sitting at room temperature on our lab bench, and all samples were well above background. And so the fact that we were able to extract protein and then put it through a sensitive and specific technology, such as the Taudia splash assay, was first one of our great wins, I'd say. Secondly, looking at this, we can see within each participant, there's some variability. Each data point is a dried plasma spot that is run in duplicate. And when we look at it partitioned out by the dried plasma spot itself with its replicate well, we start to see that there's some inconsistencies with perhaps the extraction protocol that's going on. And as we can see over on the left-hand side, the within dried plasma spot disk replicate CV was low, which was very nice, but the between disk replicate was maybe a bit above what we're looking for, and so that's certainly a point for optimization that we'll need to look into further. And then looking at p-tau17, we have a similar story with a bit more variability between the disk replicates. But again, we got nice signal above background, which was nice for a first go again, but quite the spread between the disks. However, once we take the ratio, which is our clinically relevant biomarker that we're going to look at in order to start to contribute to the diagnostic workflow of our patients, we start to see a nice trend form, which most of the samples writing below the 0.5 marker on the y-axis. And at this point, we were able to get more information from participants as the data moved through our clinic's pipeline, but we still had 4 of 16 with unknown diagnoses. And eventually, what we were able to find is that we were able to unbiasedly identify 2 of our participants as having early onset Alzheimer's disease purely from this amyloid beta p-tau217 ratio using the DPS splash workflow that we've designed, which was very exciting news. We see the different breakdown here at the x-axis, which I'll show a bit more detailed of a cohort table in the next slide. And so there are 16 participants. We had 6 cognitively normal, 2 MCI, 2 early onset, 4 behavioral variant frontotemporal dementia, and 2 PPA. And here we have also the ApoE E4 allele count for your interest as well. So grouping all of our participants by diagnostics, what we see is a very significant increase in the p-tau217 A-beta 42 ratio from the early onset AD across all other diagnostic groups, which highlights the specificity of this biomarker and the clinical relevance of this biomarker, but also helps to validate the workflow that we put together and that we are seeing what we expect to see, again, in an unbiased way, which was, as I keep saying, very exciting. And over on the left-hand side, we have the mean, standard deviation, and standard error of each of these groups. And by dividing the early onset by the cognitively normal means, what we see is a 13.6 dynamic range which is suitable for beginning to start defining a diagnostic range specific to this workflow, which will be necessary moving forward. So in summary, all data passed QA/QC. Signal was above background for both p-tau 217 and A-beta 42. The between dried plasma spot replicate well CV were low, indicating high technical precision. And however, the between DPS disk CV per participant was high, indicating that there's significant extraction inconsistencies that we'll need to optimize moving forward. The calculated p-tau 217 and A-beta 42 ratio replicates replicated the relevance of the biomarkers with known clinical features, notably distinguished the early onset Alzheimer's disease participants from all other diagnostic groups, and there were no statistically significant differences in ratio values between cognitively normal, MCI, BVFTD, or PPA groups. And lastly, which is important for our next steps, we have a 13.6 X dynamic range of this workflow, which we will hope to leverage for defining diagnostic ranges. And so in conclusion, this workflow provided a streamlined, high quality, end-to-end, globally scalable workflow. Going back to our methods, whole blood was sourced, and this was from acid citric dextrose anticoagulated whole blood, which normally we would be using EDTA or even a lancet, but utilizing these ACD tubes that we were already able to be collecting from our center shows the robustness of the Taudia chemistry, and I think importantly notes the flexibility of the workflow that can be implemented around the world, given whatever logistical constraints might be present. The Capitainer SEP10 was key, enabling a shelf-stable 10 microliter dried plasma spot storage for up to 1 month, and I imagine this 1 month can be significantly extended based on what we saw here. The elution, we were able to obtain quality protein at necessary quantity for qPCR detection. And the Taudia assay, small volume, sensitive, accurate protein detection chemistry. And lastly, the benchtop qPCR instrumentation. This is a widely accessible, cost-efficient method for biomarker detection. And then we use the Taudia software analysis for a simple, fast, and easy analysis. And so for next steps, as I mentioned, we're going to want to optimize the SEP10 extraction. We'll want to assess SEP10 dried plasma spots versus native plasma using the Taudia splash assay. And this will be important for defining any limitations of the dried plasma spot collection versus the gold standard of liquid plasma. And then we hope to expand the existing workflow to include both EDTA and lancet collection procedures for capillary whole blood. And the importance of this will be to expand the collection types to enable more flexible workflows for logistically complex sites. And lastly, we hope to define the diagnostic range to bring utility to the DPS splash workflow.
So in sum, I think we've seen from Taylor that the tools are really now available for scalable biomarker research that leverages relatively straightforward collection methods that can be really implemented not just in diverse clinical setting or clinical research settings, but potentially in home or in remote or outreach-based settings that would be more amenable to community-based research. And so this is, to us, represents a really exciting opportunity because, as I mentioned, we're really interested in trying to leverage this network that we've established with many colleagues around the world, and particularly those in less resourced regions, so that we can enable both global capacity for dementia understanding, but also clinical research related to Alzheimer's disease that really leverages this, kind of now required biomarker confirmation of disease. And so here I'm showing you just a few individuals who are part of a team that we're trying to mobilize to get funding to do a global implementation project to assess the feasibility of using DPS, basically in the field in terms of clinical research, in different contexts related to the expertise in each of these individuals, research teams, or clinics. And just to highlight, I think what the potential is for this type of workflow. I have a few quotes here from some of the individuals when they I emailed asking, hey, would you be interested in being a collaborator on this project? And many folks replied, actually everyone replied obviously, but I pulled out a few of the quotes that were particularly relevant for this talk, and so I'm just going to read two, but of course you're welcome to read the others at your leisure. And so for example, Dr. Faith Smushi in Zambia mentioned that this research is critically important, and the goal of establishing scalable Alzheimer's disease biomarker quantification is absolutely vital for settings such as ours in Zambia, which face significant constraints in cost and infrastructure. The DPS plus splash approach represents the necessary innovation required to support community representative research in the global south. Also, words from Prabha stress that in Nepal, which really like kind of made me even take, a deep breath, was the note that a project of this nature would be a new initiative in my country of Nepal and a significant step forward from our current practices. So, Prabha is a nurse, and she and her supervisor were both excited to potentially join this opportunity as basically the first initiative for doing this type of 80 biomarker measurement in Nepal, which is to me both exciting and also indicates to me that we need to do a lot of work to enhance the ability of all of our colleagues around the world to be able to help us contribute to the field of Alzheimer's disease research. So with that, I would just like to acknowledge all of our colleagues and collaborators, both at UCSF as well as Capitainer and Taudia. Of course, a huge shout out to all of the research participants, including those who provided blood samples that were part of the study that Taylor outlined, as well as to those who continue to contribute to our research in AD and related dementias. And with that, we're happy to take questions from the audience. And yeah, thank you.
All right, we do have some questions. First of all, thank you, Dr. Yokoyama and Taylor, for a great presentation. We have some questions so far, and if any do come up, please feel free to submit them using the Q&A tab. So our first question, I think Taylor can field this one. It's, is it important how you isolate the plasma? Can you use the older plasma already isolated from patients by a phlebotomy and stored at negative 80 degrees Celsius or other DPS cards, for example, or even dried blood spots as well?
Yeah, so it has to be fresh blood. That was stressed by the when I've met with Capitainer, and I think the issue is that if specifically if whole blood is frozen, there's significant red blood cell contamination that makes it through into your dried plasma spot, which can hinder the efficacy of your downstream procedures. I don't know if I can speak to if it's like already isolated plasma, assuming that the purpose would be for shipping feasibility. That would be something that I'd have to check in with the Capitainer folks.
I would guess for plasma that's collected, if it's stably transferred to a lab that could do the Taudia assay, then it would probably be very reasonable to use the splash assay to measure biomarkers in it, just as you would use any other previously banked plasma to do biomarker measures. I think the main advantage that we've found is the much lower need of input volume or concentration for the proteins themselves, which can sometimes be challenging or limiting for those that have a small volume of blood available.
All right, let's see. Our next question is, how long can dried plasma spot cards stay at room temperature?
I wonder the same thing. I'd like to know. Right now, we've done it up to 34 days, um, and we didn't notice any reduction in sensitivity or specificity in the Taudia assay. That being said, we didn't do any specific quality testing on these samples. But as it is, as long as it's not in a super humid environment, that will certainly help you out. Um, as I understand it, the drier the better. Um, but I think some longitudinal assessment would certainly be warranted before rolling this out to a much larger scale. But as of now, 34 days is what I can recommend.
And that constraint on time is largely anchored in the fact that I needed Taylor's preliminary data for a grant application to the NIH. And so we had about, I think I gave him about 5 or 6 weeks lead time, and he basically mobilized super quickly and was able to generate all of that data within a very short time frame. So I think again, we have a lot of exciting optimization ahead of us, and that is the plan as we move forward and hope that we can acquire additional funding to expand this work. But that is right now our experience is what you saw Taylor present.
Okay, and looks like we have a few more questions and they pertain to shipping. How does shipping outside the US work? I understand you can send DPS through the mail here, but curious how international transportation works.
Yeah, I can start that and then maybe Jen, if you wanna finish. My experience working with some of our different studies is that every site is different. Um, it's specific to wherever you're interested in. Every country, every locale seems to have their own regulations. Um, what I've seen is that these dried plasma spot cards are treated like standard mail. Um, I don't know if Jen, if you have anything to add to that.
Yeah, my impression, and again, I mean, I think we will need to do more research on the exact requirements for each of the local regions or regulations, but in general, the spot itself allows for isolation of the biospecimen within that component. And so, at least for US mail, USPS, once you stick it in the packaging, which is the second barrier, and then you put it in the little envelope that the package goes into, and then you put it in the big envelope where you put your address on it. That is enough sufficient, like enough barriers basically to prevent any biofluid from escaping, and so therefore is completely feasible to send in the mail without any additional restrictions. I anticipate it would probably be pretty similar internationally. I've heard from others using different types of kits that, it's also very feasible to, put these in a cardboard box and bring them on a plane back home, in the context of them being completely sealed and secure. Um, I think obviously we would need to make sure that all of the right regulatory paperwork is in place to do that. But I do anticipate that at minimum, this would mean that those of us who have colleagues who go out into the Amazon rainforest to meet families with familial forms of dementia that are living really far away from hospital or clinical settings could, at minimum, use lancets or phlebotomy to collect samples, quickly put them into the cards for storage, and then put them all in a shipping container, ice chest, and bring them back to their research labs and process them throughout the next, days or weeks, which, for many of the collaborators that we have is sometimes that's sufficient as well. Taylor, I didn't know if you wanted to continue sharing your slides or if Chris maybe has his other slide he wanted to share.
I think if you want to request control, I can pass it over to you.
I have access right now and I'm sharing, so we're good here. Uh, this next question is super interesting. So it's asking, what is the impact of samples going from wet to dry and to wet on the protein composition and stability of the sample, specifically for proteins of interest A beta 42 and p-tau 217?
I'm sorry, can you repeat the question?
Sure. It's what is the impact of samples going from wet to dry to wet on the protein composition and stability of the sample, specifically for the proteins of interest A beta 42 and tau 217?
Ah,
Oh, from like plasma to card to
yeah,
elution. Yeah.
yeah. That's a good question. I think that the biochemistry of it, maybe Daniel or Jason or if you guys can maybe comment from your perspective. I know from our perspective, um, we did our due diligence in conducting literature searches and understanding what a dried plasma spot really is, and how it can capture our analytes of interest, and everything is really analyte specific, and so everything has to be assessed in that context rather than as a plasma protein as a whole. Um, but I think what we were able to show is that we are able to capture it, and we are able to pick out um, uh, uh, early onset Alzheimer's participants out of a more heterogeneous cohort, albeit small. And so the utility that we see is there, exactly how it is affected. I assume that there's something, and maybe we can have somebody from the Taudia team help us out.
Yeah, my impression is amyloid beta is pretty sticky in general as a protein. And so, again, like Taylor mentioned, it will be a protein or target to target variability. I think some proteins are just notoriously more stable. I mean, unfortunately, that's probably also why they aggregate, right? Is they are highly stable. These, what we're measuring represent fragments, presumably from the central nervous system that are coming out into the blood. Um, and my impression is similar to Taylor's, at least for things like p-tau 217, they are relatively stable. I think the stickiness of amyloid tends to be more in the disk than for the measurements themselves. And I think that's the case for other proteins like granulins, for example. And so in that case, then the elution that Taylor was talking about, which was contributing kind of that to that variability with it between disks, is probably the part actually that was more sensitive to the targets than the antibody measurements themselves. I think once the protein got out, then it was like probably just, nicely in solution and so easier to measure. And again, that will require basically more careful measurements to understand what that variation is between targets.
Mhm. Yeah, and finding the optimal extraction buffer as well. So, if it's a tris base or a PBS base, and then we can choose a reducing agent. And you can go through the iterations that way. And that's work that I'm excited to continue, hopefully very soon.
Okay. Our next question for the percent CV, how low are you trying to get the assay down to between DPS replicates?
Yeah. The, That's a good question. Yeah, it's something that we have to define ourselves, given that this is a completely new collection method plus the assay. Um, and I always think that below 10% is favorable. Of course, if we can make it even tighter, that would be best. I think in this effort, we should always keep in mind and have our true reference of what the gold standard of biomarker collection is. And so if we can run parallel samples of the DPS splash workflow, from a same participant given like an EDTA collection, and then run that on the Taudia assay as well. That would be probably the best match and tell us what work needs to be done to optimize the DPS collection and extraction.
Yeah, I mean, presumably you'd want to see the whatever variation between duplicates you would want to be at least smaller than the difference you would expect to see between individuals of different diagnoses. And so I think the 5 to 10% or below 5 to 10% would probably be ideal. I think we in our hands don't know yet what the lower limit is for the DPS. My impression is also that the Capitainer, at least the Capitainer kit itself, is being it's also just undergone a recent um, revamp, and so we haven't tried the new one yet. So stay tuned.
Again, thank you all, especially Dr. Jennifer Yokoyama and Taylor Johnson. We all thank you for attending today's webinar. Goodbye and take care.